Okay, so it's a great pleasure to be here and to be able to share some insight into our work at GSK in bringing genetic evidence to support therapeutic target identification and validation, and some of the collaborations that we have with Bristol to try and advance science in that area. I thought by way of introduction I would introduce the team in which I sit at GSK, which is Target Sciences. Now, this is a cross-cutting team that works across a range of different therapy areas with the aim of bringing genetic evidence, computational biology, and silico bioinformatic data, and a platform of high-throughput experimental validation to try and bring evidence for the best targets and bring those targets to the right indications and to the right patients. And we realized that to do that in the highest quality fashion possible, that requires a series of both internal and external partnerships. And we recognize that the best advances, the fastest advances will be enabled through industry, academia collaboration. And why is it really important that we bring evidence to support high-quality targets? Well, it's really important because drug discovery and development is a really difficult problem. And what I show here is data on the likelihood of particular targets at different phases of the drug development pipeline being approved. And what you can see is that overall, the likelihood of any particular target being progressed into phase 1, so first time in human studies, reaching approval is something like 10% of targets. And what that means is that 90% of the drugs that we take into humans for the first time will fail. And the consequences of that are that there's huge costs in R&D per drug that's approved, and it means that these drugs take a long time and we don't get the right drugs to the right patients. And you can see that even by the time we get to phase 3, even when we get to phase 3, something like 50% of drugs will be shown not to be efficacious. So what are the sources of data we can bring at a very early stage to support drug discovery and development? Well, one of those sources is genetics. So this is some work performed by colleagues of mine at GSK, where they evaluated drugs at different stages of the drug development pipeline, so from preclinical all the way through to approved. And on the axis here, we're showing the number of targets in the pipeline at that particular phase that have genetic evidence. I realize that this isn't presenting so clearly, but what I hope you can see is that as you progress through the stages of drug development, the number of targets with genetic evidence increases greatly. And the long story short here is that for a drug that has genetic evidence to support its efficacy, it's at least twice as likely to be approved as a drug without genetic evidence. And what do I mean by genetic evidence? Well, what I mean is that there's a genomic region that encodes for a particular protein that's a drug target, and in that genomic region there are genetic variants that are associated with similar diseases. When we have that situation, a drug's twice as likely to be approved. And what that means is that in GSK last year and this year, in fact, we have an objective that 60% of our drug targets being taken forward will have genetic evidence. That's from an industry baseline of something like 10 to 15%. So how can genetics aid drug discovery and development? Well, genetics can help us identify new targets. It might pinpoint regions of the genome that encode proteins, where we believe that perturbation of that particular protein will have an effect on disease. It can also help us validate existing targets. And what do I mean by validate? Well, what I mean is that it might help us predict efficacy. So does our target have a desirable effect on our disease of interest? Could we use genetic variants to predict safety profiles? So not only our disease of interest, is our genetic variant associated with a whole host of other diseases and with adverse consequences? And maybe genetics can help us identify additional or alternative indications. Now that sounds great, and we do this routinely on a day-to-day basis, but it's worth acknowledging there are very severe limitations to the current approach that we take. And I would put those limitations into two major bins. One of which is our ability to instrument genes. And what I mean by that is find genetic variants that are proxies for perturbation of a particular target. So finding genetic variants that we think have a robust effect on our particular gene and ideally on the particular protein of interest. And it's really challenging to find those, or has been very challenging in the past. The second major limitation is that even when we have those variants that we know have a very convincing effect on a particular gene or a particular protein, accessing the large-scale data where we want to assess the association of that particular genetic variant across a range of different disease outcomes in the very largest sample sizes has been very, very difficult. And that's even for published data. So even when a study's been published, it's very difficult often to get your hands on that data in a standardized format that allows you to draw high-quality inference. And I'll give just one example of the type of study that we do here. And that particular example is on a variant that we identified through a range of sequencing studies and large-scale genotyping studies. We found a coding variant in the GLP-1R gene. Now, GLP-1R agonists are a major and important treatment for type 2 diabetes. We found a coding variant in GLP-1R that was associated with reduced risk of type 2 diabetes. Now, prior to large-scale cardiovascular safety trials being done, we could then ask, well, could we use this variant to try and predict the cardiovascular safety of this class of agents, a really important requirement for anti-diabetic therapies? The next question is, well, if we believe we have a variant here that's a mimic of pharmacological perturbation of GLP-1R and a mimic for taking this particular class of drugs, could we test it against a range of other diseases for which particular adverse safety events had been reported or for which there had been suggestions of additional or alternative indications? So cardiovascular safety first. We identified that not only was there evidence of an adverse increase in risk of coronary heart disease, we found evidence to suggest that this class of agents might in fact result in cardiovascular benefit. And this was published, I think, 3 weeks before a large cardiovascular safety trial was published that showed exactly the same thing, that GLP-1R agonists were associated with cardiovascular benefit. The next key question is, could we test it against a range of different disease outcomes and identify maybe additional indications? GLP-1R agonists had been suggested for use in Parkinson's disease or prevention of Alzheimer's. And what the specific numbers here are not so clear, but what I hope you can see is that despite the fact that we went to months, in fact years of effort to get our hands on every single individual with all of these diseases who had been genotyped for this particular variant, we really remained underpowered to make very convincing inference here about the association of this variant and thus GLP-1R agonists with these different outcomes. And I would just draw your attention, I know this doesn't present particularly well, but looking at the sample sizes we need to make these kinds of robust inference here, for CHD we have 60,000 cases and 160,000 controls. So we're talking about vast population level sample sizes that we need to do this. Now, one of the major limitations I mentioned is lack of availability of large-scale data. And I mentioned there that this particular study took us over 18 months and over 1,000 emails. Even when the data were published, that involved me and my colleagues emailing people and saying, look, you've published a paper, in all of your data there's the association of this variant with this particular disease. Can you give us those results? That took 18 months, over 1,000 emails negotiating for access to that data. Clearly, what we really want to be able to do here is to bring this evidence at the point that one of my clinical colleagues says, I think this target might be good for this disease. We don't want to wait 10 years as the drug goes through the drug development pipeline. We need approaches that are swift and give us conclusive results. So the current approach really was not fit for purpose. Looking at the availability of UK Biobank data, I think you would argue the numbers are different, but on the whole, we get almost exactly the same conclusion from those data, evidence of benefit for type 2 diabetes and for cardiovascular disease. The advance that's really enabled here is that didn't take 18 months and 1,000 emails. It took 3 hours and 3 emails to get all of this data from UK Biobank. So accessing all of those data really transform our ability here. Of course, I mentioned we want to do this widely across a range of diseases. What we're looking at here is a particular variant that we believe to be functional in the HMGCR gene, the target for statins, and we're now testing it across something like 3,000 different phenotypes in UK Biobank. Reassuringly, the strongest association that we see is high cholesterol, so we want to be able to do this across thousands of different diseases. However, UK Biobank is fantastic in the depth and diversity of data, but given that some diseases are low prevalence, even in 500,000 individuals, we often don't have the power to make very robust inference. What that means is we need to be able to access the published data and the other public data that are out there in a standard format in the way that we can here. And that's really where our collaboration with Bristol is absolutely central to our aims here, because it sounds very, very simple to say we want to access the published data and make strong inference about the role of genetic variants in a range of diseases, but it's far from simple. Other advances bring us even bigger opportunities, and with those opportunities come challenges. So recent advances in large-scale omic measurements have broadened our capacity to instrument particular genes through Mendelian randomization. I have a hard and fast rule that when I come to Bristol, I never talk about Mendelian randomization, but because everyone here knows the details far better than I do. If you don't, PubMeding George's name, you'll find hundreds of papers that will give you far more detail than I ever could. But what that means is, because now we can measure particular proteins, which are often the units that we want to target by pharmacological perturbation, we can measure thousands of proteins, and now against thousands of diseases. So that brings huge challenges. It brings us big opportunities in the sense that we want to be able to evaluate the causal role of all of these different proteins against all particular diseases, because then we can quite easily say, I think there's evidence that if we target this protein, it will have an effect on this particular disease. However, the challenge is, and I know this because I've spent many years of my life doing these things, that previously, doing a single MR study of one exposure against one outcome could take months of your life as you fight to aggregate all of that data. What that means is that if we really are serious about measuring thousands of exposures against thousands of outcomes, it needs a step change in our strategies to perform and robustly interpret those analyses. And this is where really the pioneering work by the team here at Bristol in developing MRBase allows us to take that step change. So at GSK, we're hugely excited about our collaboration with Bristol. The aims of that collaboration are both to develop the science of Mendelian randomization, to develop the resources not solely for GSK, but for the community to perform and interpret these types of Mendelian randomization studies. And of course, we're hugely excited in these types of large-scale proteomic Mendelian randomization studies about the opportunity to take those findings and translate them to novel therapeutics or better understand existing ones. To comment on the ongoing work, so we have several strands of work ongoing, but arguably one of the most exciting that we have is taking some of the large-scale proteomic measurement that's been done very recently, identify genetic instruments for over 1,000 proteins, and test the causal association with those all proteins against all diseases. And that gives us a resource where we can then, in real time, working with our partners in the therapy areas, look at robust evidence for the causal role of particular proteins against a range of different diseases that utterly transforms the way that we bring genetic evidence to support target discovery and development. And it's worth just saying this collaboration has been running for less than a year now, but the initial collaboration has already stimulated a Bristol student to come and spend several months at GSK working with some of our team there, a successful application to BBSRC for a joint PhD studentships, and 2 fellowship applications, one of which I've learned has been successful. And I'm grateful for your attention. Thank you.